Okay, we are going to talk about nucleic acids. So nucleic acids, this is the last of the 4 classes of macromolecules. Remember, nucleic acids are not used, we do not metabolize nucleic acids to convert them to ATP as an energy source, but actually ATP itself is a nucleic acid, but we don't convert it. We don't use nucleic acids in the food that we eat to form energy. So this is the one out of the 4 that is not an energy source. So nucleic acids, if we go back to the table that we started out with, the monomer form is called a nucleotide. The polymer is called a polynucleotide. And the bond is called a phosphodiester, and it's usually called a linkage, which is just another word for bond. So we are going to talk about the nucleotides, the individual monomers, the polynucleotides, and we will talk about 2 polynucleotides of nucleic acids. We'll talk about DNA and we'll talk about RNA, and then the covalent linkage is called a phosphodiester linkage. So let's talk about nucleotides first. So what is a nucleotide? So the nucleotide is made up of 3 pieces. 3 parts comprise a nucleotide. One, it has to have what's called a nitrogenous base. Nitrogenous because it contains nitrogen. 2, it has to have a pentose sugar. Five-carbon sugar. And then 3, it has to have a phosphate group. So there's different nucleotides that we will talk about. Also, you'll hear something called a nucleoside. A nucleoside is just the nitrogenous base with the sugar without the phosphate. So they're not interchangeable. You might see nucleotide or nucleoside. They actually are different things. Nucleotides are what are added to form these nucleic acid polymers. Without the phosphate group, it's called a nucleoside. Okay, so there's 2 that we will talk about. DNA, which stands for deoxyribose. So there's 2 sugars that we're going to use. DNA, the D stands for deoxyribose. That's the pentose sugar in DNA. RNA, R stands for ribose. So DNA and RNA differentiate in a few different ways. One is that DNA uses a deoxyribose, RNA uses a ribose in their nucleotides. So we're going to get into all of this. Okay, so how are these nucleotides made? So these nucleotides can be made from 5-carbon sugars, or they can be made from amino acids. There's multiple different types of pathways, and we're not going to get into the specifics of the actual synthesis of the nucleotide itself. We are going to talk about how we make DNA and RNA. So we said the difference between DNA and RNA is that DNA contains what's called deoxyribose, and RNA contains ribose. So you're not going to be doing a lot of drawing in this class on the exam. What you are going to need to do is to be able to recognize the difference. So here's deoxyribose. So here's deoxyribose. And here would be a hydroxyl. What you are looking for is right here. This is the 2' carbon. And this 2' carbon, we number sugars by the carbons. Whoops, I drew this wrong. This is not the 2' carbon. That's the first carbon. This would be 1. This is the 2' carbon. Sorry. What we're looking for here when you're differentiating between ribose and deoxyribose is this right here. This deoxyribose just has a hydrogen at the 2' carbon. Ribose has a hydroxyl group at the 2' carbon. So this is the difference between deoxyribose and ribose. And this right here, this is the first carbon, this is the second carbon. We call it the 2' carbon. So if there's a hydroxyl on the 2' carbon, it's ribose. If there is not and just a hydrogen on the 2' carbon, it is deoxyribose. So there are 2 types of bases. So we said that it has a base, it has a pentose sugar, and a phosphate. So now let's talk about the bases. So the bases, there's 2 classes of bases. Again, these are nitrogenous bases. There's what's called purines and what's called pyrimidines. So a purine looks like this. Pyrimidine looks like this. I didn't put the nitrogens in. So these are called heterocyclic. Hetero meaning there's carbons and nitrogens in the rings. So there's purines and pyrimidines. Of the purines, there are 2 different bases. There is adenine and guanine. Guanine. And then the pyrimidines are cytosine and thymine. These are the pyrimidines and the purines for DNA. RNA does not use thymine. RNA uses something called uracil. RNA only. Thymine, DNA only. Okay, so nucleosides are nitrogenous bases and the sugar. Nucleotides are nitrogenous bases, the sugar, and the phosphate. So all of these are written, all of these are pictured in your textbook. I'm not going to draw them all out. You do not need to be able to identify the difference between adenine and guanine or cytosine and thymine or uracil. You do need to be able to identify the difference between purines and pyrimidines. Okay, so the way that these nitrogenous bases, sugars, and phosphates are linked together is what's called a phosphodiester bond. And this is on slide 11. And this it has a direction. So DNA always goes from what's called 5' to 3'. And this 5' to 3' comes from the numbers, the carbon on the sugar. So you'll see this when you look at the picture. The 5' carbon is up here. This is where the phosphate groups attach. And it is going to attach to the 3' carbon of the next sugar. So the phosphodiester backbone is, sometimes it's called a backbone, is between the 5' carbon. It goes 5' to 3'. So the 3', the bottom of this, is going to attach to the 5' of the next carbon below it. I am not going to draw it out. If you look at page 11, you'll see how these are linked together, and it goes in a 5' to 3' direction. And we write the DNA in the sequence of the order of the nitrogenous bases. This would also be considered primary structure. It's just the sequence of bases. Secondary structure is going to be the hydrogen bonding between purines and pyrimidines. So a purine and a pyrimidine will always pair with each other. You'll never have two pyrimidines bonding or two purines bonding. This phosphodiester backbone is the only covalent structure in the sequence, in the polymer of nucleic acids. The purines and the pyrimidines are going to hydrogen bond with each other. This hydrogen bonding, it's called complementary hydrogen bonding, complementary base pairs. So complementary base pairs in DNA it's always going to be A forms 2 double bonds with T, and G forms 3 hydrogen bonds. So you'll see these will be dashed. So 2 hydrogen bonds between adenine and thymine, 3 hydrogen bonds between guanine and cytosine. RNA does not have any thymine. Remember, thymine is DNA only. So RNA is going to have 2 hydrogen bonds between adenine and uracil, and the same 3 hydrogen bonds between guanine and cytosine. So this DNA forms a double helix, and that there are a few different types of double helices. There's A form, B form, and Z form. I'm not going to ask you about the specifics of these. What I do want you to know is what is here. The phosphodiester backbone, this is the covalent bond that holds the polymer together. The secondary structure would be the hydrogen bonding between complementary base pairs. And you should be able to identify the difference between a purine and a pyrimidine, not draw the specifics. Also, you should be able to identify the difference between a deoxyribose and a ribose.